audible. So, yeah, once again, good evening, and thank you for joining in for this session. In the meantime, if you have any doubts from previous session or any other lectures, you can please post it in the chat box. In the meantime, I'll just share my screen now. I hope the screen is visible as well as the audio is also clear. If there is some issue, please let me know in the chat box. So I'll try to clarify if there is any issue. And in case of any doubts in between the session or from the previous sessions, please post in the chat box. We'll discuss it simultaneously as well. So this is the fifth type practice session. So this is the fifth live practice session. So in the previous weeks, we have covered about some fundamentals of applied environmental microbiology, and in this session, it will be exclusively on microbial genomics, which was the week 5 course content for this course. Uh, so based on this, the first question for today is this. So in Sanger sequencing, DNA sequence is determined primarily by what? Try answering this question. So in Sanger sequencing, DNA sequence is determined primarily by which of the following? You can just give the option number in the chat box. synthesis monitoring. We'll see a couple of more questions on Sanger sequencing and then discuss it briefly together. Okay. So in Sanger sequencing, Sanger sequencing is a method by which we determine the DNA sequence of a microbial sample or any environmental sample. So in that case, we are going to monitor the DNA synthesis, and based on that only, this Sanger sequencing is possible. So based on that, the method used in Sanger sequencing is termed as what? We will be monitoring the DNA synthesis, but based on the principle of the Sanger sequencing, the method is known as what? So in Sanger sequencing, we are going to see about how a DNA strand is getting synthesized. So that is what we monitor, and based on that, we have based on the components that we use in sequencing, we have a name for this method. You can just write down the name of the method based on its principle. Any guesses? Okay, so based on see, we are going to monitor the DNA synthesis, and it stops at one point. So wherever it stops, based on that, we find out what is the sequence of the DNA, and henceforth it is called as a chain termination method. So we have a long DNA sequence, and at some point, because of the components that we add in Sanger sequencing, the chain DNA synthesis or the chain elongation is stopped. So that is known as chain termination method. So next, a simple question: what are the nucleotides that we use in Sanger sequencing? audio clear and the screen visible, or is there any connection issue? Can someone please confirm in the chat box? Okay, so the answer for this question is so we use DNTPs. We use both DNTPs and DDNTPs. So we use deoxynucleotides and also dideoxynucleotides. So why we have dideoxynucleotides is that because they lack a hydroxyl group at the 3' carbon position. So the third carbon of the sugar usually should contain a hydroxyl group, but here that hydroxyl group is missing. So because of that, what happens? This prevents the further chain elongation. The next nucleotide cannot come and bind to the chain through this sugar because of the uh, lack of hydroxyl group here. So, and that is where your chain termination happens. So all the three questions that we saw now about Sanger sequencing is based on the use of this DDNTPs or the dideoxynucleotides. Next question again based on Sanger sequencing. So there are a lot many questions based on Sanger sequencing because that was the one of the first used sequencing methods. So it's very basic. So if you are clear about how the sequencing works and stuff, then it will be easy for you to understand the other or the more advanced sequencing techniques. Okay, so now the question is the main enzyme component of Sanger sequencing is what? Do you know about how this DNA chain elongation and all happens? Then this question can be answered. So even in, if you know about PCR, we have discussed about PCR as well. So if you know about PCR, even then this enzyme is used. A different form of this enzyme, but yeah, it's the same enzyme and the same activity that happens. So the main enzyme component of Sanger sequencing that helps in the chain elongation is what? Anyone wants to take a guess? Don't bother if it is right or wrong. Just try to post your answers in the chat box. This is just a practice session. So I also know that you are in the meeting. And if it is two-way, then it will be good because the session is about DNA elongation or in the replication process in PCR process you have studied, then this is the enzyme that facilitates the addition of nucleotides to the DNA strand so that the chain is formed, the polymer is formed. So it is DNA polymerase. Okay. And this question we have discussed in one of the previous questions. So I think this is easy. You can just post your options in the chat box. What is the primary function of DDNTPs? That is dideoxynucleotides in Sanger sequence. Why do we use them? Just a few minutes back, I just told why we use them. Can you just write out in the chat box? People who are in a meeting, because this question we have just discussed a few minutes back. Can some of you just post the answer in the chat box? Okay, so the answer for this is the termination of DNA chain elongation. So that is why we use DDNTP, so that the chain further does not elongate because it lacks a hydroxyl group at the third carbon position in the sugar base of the nucleotide. Okay, so this is the overview of Sanger sequencing. So it works based on the chain termination method, where we add a lower ratio of uh, normal nucleotides with DDNTPs in the reaction mixture. So what happens is because the DDNTPs lacks the 3 prime OH group or the third carbon OH group, so the further formation of phosphodiester bond at this point is like stopped. So therefore the DNA polymerase incorporates DDNTPs at random, but after DDNTPs the chain elongation is stopped. So once after the PCR is done, we get billions of oligonucleotide copies of the DNA sequence, each stopped at one point. So this we can separate it using a gel electrophoresis method. So we use gel electrophoresis usually as a separation technique. So based on the size of the molecule, the separation happens. So because each of the strand is terminated at a particular point, one would be shorter, one would be longer. So that is how you get a pattern out there. So they separate based on wherever they stopped, uh, right? So and then you can find the pattern, and we can read the gel and then try to um, figure out the sequence of the DNA. So this is how Sanger sequencing works, and this is one of the very basic sequencing techniques. If there are any doubts in this technique, you can just post in the chat box. Otherwise, we'll just go to the next question. So since we have seen the basic of next Sanger sequencing, next we'll move on to some advanced techniques. So the next question is about NGS sequencing, which is the next generation sequencing. So NGS consists of dash steps in sequence. This is an open-ended question. So based on my mind, I've prepared the question. Whatever you know about NGS, this is just first question to get introduced with that. So yeah, you can try answering this. Okay, so the answer for this is NGS consists of three main steps, which is library construction, amplification, and then sequencing. So first there will be some kind of a library construction that is happening, then you go for amplification, and finally the sequencing. This is the sequence of how a NGS happens. Okay, next question is about ion torrent semiconductor sequencing. What do we do in ion torrent semiconductor sequencing? In simple, whatever is your understanding, you just post it in the chat box. can just tell the principle in a few words, like 2 3 words. Okay. So in ion torrent semiconductor sequencing, basically the principle is the pH difference. So we'll discuss more in detail after a few questions. Oh, so which of the following does NGS employ? NGS stands for next generation sequencing. So we saw what was the sequence of NGS a couple of questions back. So here the question is, which of the following does NGS employ? I hope the screen is visible and I'm audible. Uh, is there any issue with the audio or the screen being visible? Can I get a few thumbs up if the screen is visible and the audio is clear? Okay. the correct answer for this is sequence by synthesis principle, and chain termination we use it for use it in Sanger sequencing where we use this TD and TPs, so that once the TD and TP is incorporated in the DNA strand, there is termination of DNA synthesis. We'll just see the theory of all of it in a few slides. So we'll just first solve some questions and then see the theory like we saw for Sanger sequencing. The next question is about bridge PCR. So which of the following does bridge PCR promote? Okay, so the answer here is clusters of identical strands and not ligation. Ligation means joining of two complementary strands, right? So that we are not essentially doing in bridge PCR. What we do in a bridge PCR is there will be DNA fragments which we have to sequence. So typically bridge PCR is used in NGS, right? So here what we do is we have DNA fragments. We try to attach DNA fragments with adapters at both the ends. Now one end of that adapter inserted DNA is attached again to a solid surface, and there is one more end which is the free end. Okay, so this free end can bend over and hybridize with a nearby complementary primer. So there will be a primer also there in the system. So we have a DNA segment with two adapters. So one side of the adapter is fixed to a solid surface, and on the other side it is free. So when it finds the complementary surface, now this DNA fragment bends and then anchors, and henceforth we call it a bridge PCR because it forms a bridge kind of a structure. So after this, we have the DNA polymerase coming with the picture again to synthesize the complementary strand. So because we already have the primer here, it's just the DNA polymerase that has to come and synthesize the strand. Like this, we have this process happening repetitively, and hence a cluster of identical strands are generated. So only when there is enough number of clusters we have, then we have the strong signal for the sequencing detection process. So here technique not to use fluorescence in camera scanning. Ion torrent sequencing was the first commercial technique which did not use fluorescence or camera scanning. We saw that it uses pH differences, ion torrent semiconductor sequencing. So yeah, this we saw ion torrent sequencing detects nucleotide incorporation through pH change from proton release. The next question is during pyrosequencing, detection of nucleotide incorporation occurs through which of the following? So we have seen about Sanger sequencing, next generation sequencing, ion torrent semiconductor sequencing, and now we have pyrosequencing. Exactly. So in pyrosequencing, whenever there is a nucleotide being added, it releases a pyrophosphate, and this is then converted to ATP by sulfurylase, and so this reaction drives the conversion of luciferin to oxyluciferin with the help of a luciferase enzyme. was to use in pyrosequence. The enzyme used in pyrosequencing is, I just now pronounced it. Exactly, it is Leucine. Okay, so next we talk about nanopore sequencing, where from your perspective, what is the advantage of using nanopore sequencing out of others? Nanopore sequencing, the primary advantage is that it allows the sequencing of very long DNA fragments. So apart from that, that is the main advantage of nanopore sequencing. Technically, when comparing it to other Sanger or NPTEL, and also uh, there are advantages that it allows real-time data analysis. It is also a portable process, and it's kind of cost effective, and you need only very less sample compared to others. So in some situations you have a very less number of samples, like I mean less amount of samples. So in that case, this is very advantageous. The primary advantage is that it can do ultra long reads. Apart from that, it is portable. It allows real-time data analysis. It's cost effective, and it can also allow less sample requirement. separation of two DNA strands. Like when we talk about in the case of DNA context, it it refers to separation of DNA strands, that is known as denaturation. Yeah, it occurs in the process, maybe in one of these steps, but when we talk about sequencing methodology, then it is called as shotgun sequencing here. So next we'll try to solve assertion reason question. So the assertion here is the Sanger sequencing can read longer fragments than many high throughput methods. Reason is it sequences DNA using chain termination during synthesis. You have to tell whether both the statements are right or wrong, or if one of it is right and the other is wrong. Exactly. So Sanger sequencing, we can read a longer DNA fragment because it's all about chain termination at one point, and then we trying to do a gel electrophoresis and elucidate the sequence. So yeah, it can read longer fragments than many high throughput methods. High throughput methods, what we mean by that term is that it can sequence a large number of samples at the same time. So in Sanger sequencing, yeah, the process can be slower, but it can read longer fragments than high throughput methods, which can process large number of samples but not as longer. the reason why third generation, which is electrochemical based sequencing, is cheaper than the second generation or the optical based sequencing system. So here we have that next generation sequencing steps. So first is the library preparation. So the DNA is fragmented using enzymes or physical methods, and then they are ligated to platform specific adapters. So the next, the template DNA is amplified using a droplet or solid state PCR, so that we get number of copies of the template molecule, and then we take it to the actual sequencing itself, where we try to find out the specific nucleotide is there in the template. So for this we use optical systems or electrochemical systems. This is basically the next generation sequencing. And finally, you are trying to record the signals, and you do the analysis and find out what is the sequence, and after that you try to compare it. All these comes into data analysis. So we have different types of PCR that we use for this. One is the emulsion PCR. So this emulsion PCR is generally used in pyrosequencing, proton sequencing, or sequencing by ligation. So first what we do is we take the adapter ligated template. We have a DNA to which you have attached an adapter. You take primers, polymerase, and beads, everything within missile. Okay, missiles are small round structures that you can see, like these round structures. When you zoom into it, you can see that you have the adapter ligated template, primer, polymerase, and beads. So all these are present within the micelles. So the template binds complementary primers on the bead, and further the PCR is generating the complementary strands, because we have all the essential nucleotides as well as polymerase on it. So initially you have a template attached to an adapter, and then it binds to a complementary primer, and then the strand synthesis starts. So beads with clonal polonies are then used in sequencing. Yeah, this is the basic of the sequencing. And next, when you talk about bridge PCR, this is the next called, is used in investigating short reads, alterations in our genes based on our behavioral and environmental factors is what we study in epigenetics. It's again a fascinating branch where we try to correlate between what we see and how genes work. Okay, this is the preface of the next topic kind of a thing that we'll be seeing. So, what analysis do we do to identify various microbes in an environmental sample? Okay, so ideally, what we do is a microfill analysis, but in this context, again, we try to do metagenomics. Meta is an umbrella term. So when you talk about metagenomics, you do it particularly for environmental samples because of the diversity of the organisms that is there. You have n number of microorganisms in one sample, and you cannot individually try to identify, sequence and identify those organisms. So essentially, what you do is a metagenomics analysis. What does the term methylome refers to? what are the environmental or behavioral factors that cause some genetic change. So one of the change could also be methylation. So DNA methylation again turns off genes. So there is something called turn off and turn on of genes, where we try to refer about how the gene is being expressed, whether the expression is enhanced or whether the genes are turned on. A gene that was not expressed before could be turned on, or a gene that was being expressed could be turned off. So DNA methylation is one way where the genes get turned off, and when you try to study the complete set of DNA methylation modifications of a cell, then you call the process as methyl. Then you call the data complete set of data, and this is referred as methylome. So in genomics, what you have is you have the microbiome. You collect the sample, extract the DNA, send it for sequencing, but all the DNA that is present in the sample is being sequenced. So the data that you get out of it is the metagenome. So in DNA, you can study different omics, which is genomics, metagenomics, epigenomics, and metallomics. When you study about RNA, it is called transcriptomics. When you study about protein, it is called proteomics. When you study the process of translation and this translational dynamics, then you call it as transcriptome. And when you study the interactions, it is interactome. When you can, apart from these three molecules, which is DNA, RNA, and protein that you see in the central dogma, you can also study about various metabolites. So this includes metabolomics and glycomics. And finally, when you study the organism itself, you call it as microbiome, like you told, microbial analysis, microbiomics, viromics for viruses, and microbiomics for fungi. So the next part would be about some bioinformatics. So that it is again a simple question. The question looks very complicated, but just think for a second and you will get the answer. Dash generate alignment between a nucleotide or a protein sequence. You call that as a query, and a nucleotide or a protein sequence within a database, which is referred to as subject. So you give a nucleotide or protein sequence called as a query to something, and it again tries to match it with a nucleotide or protein that it already has in the database, which is known as subject sequence. So where do you do this kind of alignment? form of that too. Okay, so in this process we use different file formats. We'll see some questions on that. So what is the difference between FASTQ files and FASTA files? Okay, so the answer for this question is that just quality scores for each. So in bioinformatics we use both FASTA files and FASTQ files, but the FASTA file is a text-based format. So here you have the nucleotide and protein sequences, but in FASTQ files what you have is, in addition to the sequence, you also have a quality score that uh, is present. So not only the sequence, but also you have a quality score here, and that is the major difference between a FASTA file format and a FASTQ file format. Okay, based on that, we again have a match the following question. You can just try this question. saw in half of bioinformatics. So in BLAST, it is a basic local alignment search tool, and we can perform different types of alignment searches here. So based on that, we have different types of BLAST. So in BLAST in, you are going to give a nucleotide sequence as input, and the search is going to be in a database which contain nucleotides itself. So where you use this type of BLAST is for sequence identification, you use BLASTIN. And next comes BLASTX, where you are going to input a nucleotide. The tool itself will convert your nucleotide sequence into a protein sequence. It will itself, whenever when your nucleotide sequence is going to be translated, whatever the protein sequence will be correspondingly gotten, so that will be done by the tool itself, and then it will try to align or match it with a protein database. So this is BLASTX. And next you have BLASTP, where you enter a protein sequence, and the search is happening in a database of a containing protein. And next you have TBLASTX. Here your query sequence is a nucleotide. You are going to input a nucleotide, and it is going to be translated into a protein. Again, there will be a database of nucleotides that are, again, the data is translated to a protein data and being stored. So you are entering a nucleotide sequence, the database also has a nucleotide sequence. Both these sequences will be tried to converted to a protein sequence. So whenever translation happens, whatever protein sequence correspondingly we will get, it will be uh, changed, and then these two will be tried to be aligned, and then you will get your result. And finally, when you talk about T-blastin, you are going to input a protein sequence, and then you in your database you have only nucleotides. Those nucleotide sequence will be tried to convert into a protein sequence, and then these will be aligned and searched. So these are the different types of blast you can perform. Again, a easy question. We have seen it in 2-3 questions before. Which sequencing generation first move away from optical detection systems? Just wait for 1 minute to get the answer for this. this. So emulsion PCR, you have micro droplets or micelles, and amplification happens based on that. In bridge PCR, you have surface bound amplification, and in clonal amplification, there is colony growth, and in standard PCR, you just do it in a single reaction tube. This is the pattern. So this is a typical step in a DNA microarray. You have different cells, you culture them, you isolate, do the RNA isolation, and then you do the reverse transcription. You get the complementary DNA or the cDNA, and you do the fluorescent tagging, and you hybridize that onto a microarray. So based on the tags, you can detect the tags that you have. So this is a typical microarray process. You can customize it based on your application. Okay, in the next part we'll see some other genomic topics. So the gene transfer from one bacteria to another that is not its offspring is called what? Okay, yeah. So plasmid gets transferred from one organism to another, which is not an offspring. It's from the same generation itself. The plasmid gets transferred from one microbe to another. That is true. Transposons as well equally true. But in this context, I was not asking what is getting transferred. I mean, what gene is getting transferred. The gene transfer process in itself is called what? And that is horizontal gene transfer. Of course, your answer was right, but in a different context. What are the three modes by which horizontal gene transfer can occur? So now we have seen that the gene transfer from one organism to another, which is not an offspring of, is called as horizontal gene transfer. And typically plasmids, transposons, all of these can get transferred. So these two we have seen. Now the third aspect is, by what mechanism does horizontal gene transfer occurs? happen via transformation, transduction, and conjugation. So this is what we call as horizontal gene transfer, and it can happen via three modes. I'll just skim through the remaining of the questions, the question and answer as well, because we are running short of time. So the next is about the DNA sequence. Uh, so sorry, the codons, which is a nonsense codon, missense codon, and silent codon. So a nonsense codon gives you incomplete protein, whereas a missense codon gives you a faulty protein. And the silent codons give you normal protein. And next question is about different agents that we use. One is acridine, 5-bromouracil, and UV light. So acridine is basically an intercalating agent, and 5-bromouracil is a base analog, and UV light causes thymine dimers, and all these three agents are in relation to gene mutation. So 5-bromouracil is a base analog, and UV light can cause the formation of thymine dimers, and acridine is an intercalating agent. All these causes gene mutation. So here you have a complete set of mutagens and what they basically do. So in transduction process, the genes are transferred from donor to recipient through some agents, and these agents are usually phages or the bacteriophages. So a bacteriophage infects one bacteria, gets a part of DNA from the bacteria, and when it is going to infect other bacteria, it's also going to transfer this part of the DNA to that bacteria. So this is the basis of transduction process. And the next question is gene present in different species due to speciation from a common ancestor is called an ortholog. Okay, this is in another slide explaining conjugation, transformation, transduction in detail. I think it is misplaced. So look at it afterwards. You just have a look. So in conjugation, you basically the cells use a contact through pilus for the gene transfer. In transduction, we saw bacteriophagic cells in that, and in transformation, it is kind of a direct uptake where free DNA that is present in the environment is directly uptaken by another bacterial cell. Okay, next is kind of an application based question. So where we use what? In case of wastewater surveillance, we try to use population infection trends, we try to find out. And when we talk about genome epidemiology, it is used for tracking the transmission of diseases. And next, when we talk about resistance gene monitoring, we talk about environmental reservoirs where this resistant genes are there and from where it is getting transferred. And when we talk about pathogen sequencing, we try to monitor what are the variants that are present. We try to detect them and bring about some measures. Okay, this we saw in previous, the Shannon index, all of these things we saw in the previous session itself. So alpha diversity essentially means the species diversity within one sample. So these are the questions for today. I hope the session was useful. If there are any comments, you can let me know in the chat box, or if there are any doubts, please post in the chat box. Otherwise, thank you for joining in for the session. Hope you got some learning from the session. Thank you for joining in.